new perspectives in the management of age-related diseases. And I think you're really in for a treat because we have a very interesting program, but we also have some fantastic speakers that will be joining us here at this session. What you will be hearing is an update on the prevalence and the cost of neurodegenerative diseases. We'll also hear about the latest advances in research and management, and the whole session will have a particular focus on Alzheimer's disease. As you will see from the agenda, we will hear from 3 eminent experts to learn more about the causes of the disease, the role of age as a main risk factor, and we will receive a very interesting update as well on how an innovative therapeutic approach called AMBAR therapy, which is a plasma exchange with albumin replacement, how that can be implemented in a real-world setting. Our speakers will have Professor Andrew Scott. He's a professor of economics at the London Business School, and he's also a co-founder of the Longevity Forum. And if everything goes well, we'll actually have him joining via Zoom here today, and you can actually see him on screen. So very nice of you to join us. Great to have you here.
Hola. And,
hola, even. And then the next speaker will be Dr. Steve Braithwaite. He's the Chief Scientific Officer at Alka-Hest, which is a US-based Grifols company, which is very active in Alzheimer's research, but he's also an adjunct professor of neurology at Rutgers University in the state. And then last, but in no ways least, I'm really very happy to be able to introduce Merci Boada, who is not just a very internationally renowned neurologist and researcher, who is very active in European research projects and network. She's the founder and medical director of AFE Alzheimer Center here in Barcelona, but also from my side, she's really a very dear friend and an advocate on behalf of people living with Alzheimer's disease, both in Europe, but also globally. And something I completely forgot to do myself is actually introduce myself. So my name is Jean-Georges, I'm the director of Alzheimer Europe, which is the umbrella organization of national Alzheimer's organizations in Europe. But just before we start as well this session, some kind of housekeeping rules so that we know how to interact. Something that the organizers of this session would really like to hear from you is how interesting, how useful did you find the session that we are organizing? So please use that QR code, which will get you to the feedback form, and please let us know what you think of the session and the speakers and the organization of the conference. Also, I would imagine that all of you have already downloaded the conference app, so please use the conference app to ask questions. It will not be possible just to raise your hands, so please use the conference app to ask questions. And on the conference app, you can click on the question and answers tab on either your home screen, and you select the right session. And if you connect remotely from your PC or laptop, because people are also joining us remotely, you can also ask questions via the conference app. And we hope that we will find some time at the end of the presentations to address your questions. But now, without further ado, I would like to invite Professor Andrew Scott to start this symposium with his presentation entitled Aging as a Dominant Risk Factor in Neurodegenerative Diseases and its Socio-Economic Implications. And with that, over to Professor Scott.
So thank you, Jean-Georges, for that introduction. So yeah, I'm an economist who focuses upon longevity. And my research is about how we seize the advantages of longer, healthier, and more productive lives, and how we seize the economic benefits of that in terms of GDP and employment. But also, and what I'm going to focus on here today, is thinking about the value to us in terms of dollars of health gains from aging better. So that's going to be my focus here today. Now, the normal way of approaching this topic is to talk about an aging society, the fact that there's now more old people than young people. The birth rate's falling, people are living for longer. So now every country around the world is seeing an increase in the number of people aged over 65, over 80. And in fact, centenarians is the fastest growing age group in the world. So now, for the first time ever in human history, there are more people aged over 65 than under 5. But I think that's not the best way of looking at this topic. So rather than look at an aging society, looking at changes in the age structure of the population, I prefer to look at longevity and changes in how we're aging. And the beginning here is this extraordinary chart called showing you best practice life expectancy. This is the country at any point in time with the highest life expectancy at birth. So currently that's Japan, where Japanese women have a life expectancy of 87. And what this chart shows you is over the last 100 odd years, in every decade, life expectancy has increased by 2 or 3 years. So every generation has been living 6 to 9 years longer than the previous. And it's how we seize the advantages of those longer lives that my research focuses on. And in particular, there's 2 aspects that are critical to longevity. The first is recognizing that ultimately it's not about having more old people, but having more time. If we live longer, we have more time. And how do we use that time? So if you ignore the weekends and give yourself the nights off, everything else, let's call productive hours. If you live to 70, you have 125,000 productive hours. If you live to 100, you have nearly 100,000 more hours. What do we do with that extra time? And that's not just about the end of life. It's not just about retirement. We're going to have to work for longer. So therefore, we spend more time developing as adults, and we get married later, and we have children later. We do more education. We have mid-career breaks. We have shifts in our career. And that's very much the focus of my book, The 100 Year. But as well as using all that extra time, not just at the end of life, but throughout life, there's also another key feature of longevity, which is the subject of my focus today, and that's malleability. We know that how we age is malleable. We know that exercise and diet, the environment, sleep, our sense of purpose all influence how we age. And increasingly we're aware that chronological age does not reflect biological age. And that, of course, is why we are living longer and, on average, in better health. But, of course, the longer we live, the more important it is to exploit that malleability of age. And this chart here shows you all the more why that's important, because the lines at the top, the blue, the orange, and the grey line, they're what are called survival rates. They're for the United States, and they show you the probability of a child being born in 1933 or 1975 or 2018 living to a certain age. And the area under those curves is life expectancy. So you can see between 1933 and 1975, big increase in life expectancy, but it came mainly from improving survival rates in the early years or in midlife. If you look at the shift between the orange and the grey line from 1975 to 2018, you can see the life expectancy gains are coming now much more at older ages. In fact, for most of the rich countries, more than 50% of life expectancy gains are now coming at ages 70 and above. And that green line you see in this picture here shows you how frailty and disease accumulates with age. So increasingly, life expectancy gains are happening at a time of life characterized by frailty and illness. And that, of course, has led the shift from infectious diseases to non-communicable diseases. What, of course, is striking about those non-communicable diseases is just how strong related they are to age. They are age-related diseases. And, of course, COVID has dramatically changed the mortality rates, or at least death rates, but COVID itself is unusual as a virus in the sense that it so mimics the mortality rate effect with age that you get from all other causes of death. So COVID in some sense is another viral form of aging. Now, of course, given that importance of age as a sort of underlying factor behind so many diseases, there's been extraordinary biological developments in beginning to better understand biological pathways of aging and manipulating those in the lab. And there's been a number of books, great books written explaining those scientific developments and the excitement that they can potentially bring. And of course, on a topic like this, no end of newspaper articles. We've also been introduced to C. elegans worms, which are the subject of much manipulation in the lab, as well as creatures such as the naked mole rat, who don't age. But I want to focus not on the science, but on the economics. And again, I stress this is not about how longevity affects GDP, which is a huge topic. But this is about the value of health improvements to us. It's about using the economic tools we've got to evaluate health gains. And these tools are used across a variety of purposes to help governments allocate budgets. What are the most pressing diseases? What are the most valuable ones to try and approach? And at the heart of these economic tools is a notion called the value of statistical life. And that statistical life depends on two different features: the quantity of life, life expectancy, but critically also the quality of life. And in the economic models that I use, that's captured by three phenomena. One is your health. Quality of life is higher, the higher your health. The other is your consumption, your standard of living, but also your leisure. So we're going to think about how, if I change life expectancy or if I change health at any given age, how does that change my life, both in terms of quantity and in terms of quality? And because this is an economic model, if I'm living for longer, I know I have to work for longer. So there's all these trade-offs that are going to happen. So as we change health and we change life expectancy, people make different economic decisions. They make different work plans, they make different financial plans, and they consume different amounts. So we can then think about these two different lives, one with, say, current life expectancy and current health, and another one with a different life expectancy and a different health. And we can put a dollar value on how individuals value the change from one of those lives to another. I say, not about GDP, it's literally about how we value health itself. And of course, talking in still with a shadow of COVID hanging over us, it's pretty obvious the last few years that health is really valuable to us. We've seen governments around the world take actions that lower GDP to save lives. So we know that health is valuable, but let's use these tools to think about how we would value improvements in aging. This results are drawn from a recent Nature Aging paper I had with colleagues Martin Ellison and David Sinclair. I'm going to think about 4 different cases. One is we call Strulberg from Gulliver's Travels. This is a world where we just increase life expectancy, but we don't improve how we age in terms of health. Health continues to deteriorate with age. Another one is Dorian Gray, which is the very opposite of Strulberg. Here, we're not going to extend life expectancy. We're just going to improve health at every age. So we're going to improve the rate at which health deteriorates with age. There's another one which is called Peter Pan, where I'm actually going to slow down aging itself. And by slowing down aging, we get 2 benefits. We slow down mortality, so we live longer, and we slow down frailty, so we're in good health for longer. And then there's a final one called Wolverine, where we actually reverse aging, and you go back to health of a previous time. Lots of results in the paper, lots of numbers. Let me try and give you some of the punchlines. First of all, just extending life, it's valuable. Life is sweet. But of course, if all we do is extend life but not improve health, it hits diminishing returns because you're living ever longer in ever poorer health. Much more valuable is Dorian Gray. That is not to extend life expectancy, but to improve health. In fact, we can show that a full compression of morbidity, getting healthy life expectancy to be the same as life expectancy, is more valuable than any future improvements in life expectancy itself. We've reached a point now where the most valuable thing for us is to compress morbidity, to make health span match lifespan. Of course, best of all is to slow down aging, that Peter Pan example, because there you get 2 benefits. You do get longer lives, and you get it in better health. And those 2 together give you what I call complementarities in economics. I don't like living for longer in poor health, but if you can improve my health, I value living longer all the more. Now, just a flavor of some of these results. These are big numbers. Remember, health is really valuable. So you can see that for a 60-year-old, 1 more year of life, but without touching health, is worth $286,000 in the US, according to kind of standard calibrations. But notice how that number diminishes. The Dorian Gray is putting a value on improving healthy life expectancy, not life expectancy. And just look how big those numbers are compared to Strulberg. It is incredibly valuable to age well. And of course, that makes sense because if children born today in rich countries will live to be 90, the most pressing health challenge is to be healthy when you're old. Now, let's try and dig a little bit around about why these numbers are so big, and I'll get to even bigger numbers later. the gains to treating a disease depend upon the probability of someone experiencing it. So given now, for the first time ever in human history, a child born today is expected to get old, then that puts a huge value on tackling age-related diseases. When you only had a 1 in 6 chance of making it to 80, treating age-related diseases were not seen as valuable. When everyone has a more than 50% chance of reaching 90, it suddenly becomes very valuable. The other thing is, economically, the value of treating disease depends upon how soon you're going to experience it. So as the aging society happens, the value of treating aging gets bigger and bigger and bigger. And of course, as we saw, age-related diseases are now the most important set of diseases. So the other thing I want to try and show you is how targeting aging itself rather than single diseases gives huge benefits in terms of the individual and their willingness to pay. And it really comes about from 2 pretty obvious factors. The first is, if you can slow aging, as in the Peter Pan case, then of course you're going to affect the incidence of multiple diseases. So as opposed to just tackling dementia or just tackling cancer, if you can slow down biological aging, you're going to get a benefit across very, very many diseases. So that's already going to be a large number. But the other thing that's really important is the synergies that are unlocked. Because if we could, for instance, get rid of cancer, that's great, but there's still dementia or cardiovascular diseases lurking around. So the quality of life is not improved that much if you're with cancer, if there's still a threat of those other diseases. If you can slow down aging, you reduce the onset of all the diseases, and so you get the synergies from that, which make this number so large. So tackling aging rather than single diseases leads to these huge gains because you're aggregating and you're unlocking synergies. Now, to show that, we play around with some examples from using metformin. Now, I'm not here to say metformin is a great anti-aging treatment. There is a study that says this is the impact of metformin, so we just use that. Now, the point I'm interested in is metformin is not itself, but as a potential anti-aging treatment. And this shows you the value to the individual of an anti-aging treatment compared to completely eradicating cancer, completely eradicating dementia, or completely eradicating cardiovascular disease. All theoretical options. I'm just trying to value them. And you can see that the gains are huge to the individual for all of these, but you'll also see that actually just anti-aging treatments give sums that are greater than just eradicating single diseases. Those 2 things that I was talking about. And here I'm just trying to show a chart to explain the value of this, because this is the value of an anti-aging treatment in reducing cancer, dementia, cardiovascular disease, and frailty. And if you sum across all those single diseases, you get the blue line. But the great thing about anti-aging is not just it sums across all those individual diseases, but it unlocks those synergies of better health across the board. And those synergies are captured in the difference between this black line and that blue. So if I've just looked at the individual, I want to now think about society as a whole. Now, of course, we've seen some big numbers. I've shown you numbers of $300,000, $400,000, $500,000 for the individual. So you can imagine when we go to the population, we're going to get very big numbers. But I'm also going to show you that something's different about aging. When you start to focus on aging as a treatment, you're going to get very different results from thinking about other diseases. And here's the answer. This is the US population in 2017. And this is the projection, the solid line is a projection of the US population on current mortality trends. If you could improve aging so people were healthier for longer and lived a little bit longer, then you get all these additional people living. Which of course means you've got more old people, which means the value of treating aging increases. In other words, there's a virtuous circle around aging. The better we get at aging, the more we value further improvements. So what we did, we tried to calculate the aggregate gains to the US of slowing down aging. Slowing down aging, so you just got 1 more year of life expectancy and an improvement in healthy life expectancy. And the numbers are staggering. We find that just that 1 year increase in life expectancy by slowing down aging leads to a huge multi-trillion dollar. These are $72 trillion of gains. 1 more year of life expectancy from slowing down aging, we calculate is worth $38 trillion to the United States in present value terms. Health is incredibly valuable. What is the most pressing health challenge? Age-related diseases. Therefore, tackling aging leads to these multi-trillion benefits. But what's different about tackling aging compared to other diseases is the better you get at aging, the more you want to value further improvements. When people are frail in their 80s, people don't want to live in their 80s. But if you can get them healthy through their 80s, then they want to live into their 90s. All past diseases, the better we've got at treating the disease, the less interested we are in treating that disease. The better we get, the less interested we are in further gains. When it comes to aging, the better we get at aging, the more we value further improvements in aging. In other words, because now, for the first time ever in human history, a young person can expect to live into old age, the most pressing high-income demand in the health system is to age well. That starts well before people are old. It's best achieved by tackling aging. The benefits are multi-trillion dollars because you're aggregating across many diseases and unlock the synergies. But the better we get at aging, the more people will value even further improvements in aging. So this is a really radical change, not just in humanity. For the first time ever, the young can expect to become the old, but also in our health system, which is now going to focus increasingly on achieving healthy aging. So it does seem to be different this time. Of course, we don't yet know what progress the biology of aging will make in coming up with workable treatments, but the value of coming up with workable treatments is just going to increase the more old people there are and the better we age. And given this new human imperative of aging well, this is a market that can just be expected to grow and grow. Society is already beginning to adjust to these gains in life expectancy. The mistake is always to think of it as about old people, but it's really about changing how we age. The most important market is not the silver economy, looking after old people, but the evergreen market of helping us age well. People will be prepared to spend money to be looked after in old age, but they spend even more if they can be helped to age better. So this is the market that is beginning to emerge. It really is an issue of all's well that ages well. I hope that's given you a flavour of some of the results from that paper. If you're interested in more on the social and economic issues, then there's a couple of books I've written, and there's ways also you can keep in touch with me in the future. Thank you.
Thank you so much, Andrew, for this really thought-provoking presentation. I think it was really interesting how you mixed concepts of aging, longevity, introduced economic arguments, but also really made us think using examples of literature, pop culture. It's not every day that I hear presentations where we're mentioning Peter Pan, Wolverine, Dorian Gray, and Gulliver's Travel. I really thought that was a fantastic presentation. Also the idea that we shouldn't just think about combating diseases, but really combating aging in itself. And from that, I'm moving on to our next speaker. It's Stephen Braithwaite, and his presentation will be on the plasma proteome and the implications on aging, and also a therapeutic potential of that plasma proteome. Stephen, over to you.
Thank you. Thank you very much. And move over here. So it's great to be able to build on many of the concepts that were just brought up by Professor Scott, and really go into the science of aging. Can we really make this impact? The big picture sort of goal is very clear, but how much is there reality in this area? So I just want to start off sort of thinking about this. When you ask people what is aging, you often first come up with the chronological aspect. You know, it's important, but it's not getting to the biology. Then people move on to, well, it's wrinkles, it's baldness, these sort of concepts. But we really need to understand the biology. And if we can understand the biology, potentially there are therapeutic approaches which can help us target aging biology and get to this point of not thinking just indication by indication, but fundamentally target the biology of aging and have this broader impact. So I'm going to give you some examples today in how we've been approaching this, understand the biology of aging, and then some therapeutic approaches to show you there really is reality to targeting this biology. So what is the biology of aging? So we know quite a bit now about the biological process, and if we understand these biological processes, we can start thinking of therapeutic approaches. Clearly, aging is complex. It's multifactorial, and many groups have studied into this biology and started to understand, here are the mechanistic components. And these particular components are things like immune aging, inflammatory aging, inflammaging, cellular senescence, epigenetic changes, genomic changes, protein handling changes. We're starting to understand this biology. So it's complex, but we've got a handle. We've got a starting point of how we could approach the biology of aging for treatment-type purposes. So this has also come sort of in line with a lot of our interests in how would you go about attacking this multifactorial biology. Are there ins to this that are very clear to us? And I've long had interests in the plasma proteome, and when you think about plasma and blood, you see in plasma the representation of a very broad biology. You see genetic changes, you see effects of diet, you can look at lipid changes in the blood. As we exercise, if we're obese, we can see the markers, we can see biomarkers of changes of life. And so you see this as pregnancy, aging, diseases, it's all there represented in plasma. So maybe this is a place where you can actually see, here is the biology, a broad multifactorial biology which is represented, and maybe a source to look at. And when we think of blood, it connects all tissues. And when we think of aging, you don't just age one tissue at a time, you age systemically. So every organ, the brain, the lungs, the heart, everything ages. And if we need to think about aging, we need to think in less of a, here is one very specific target. Here is something more systemic that we want to change. Blood may be the source to think about in this biology of aging in a very defined way. So clearly we use blood a lot to look at biomarkers. We look at this as a readout about biology. Some really big advances have been shown by looking at functional effects of blood and plasma across a range of organs. And I got particularly interested in this when some publications came out from the lab of Tony Weiss-Corre at Stanford back 10 years ago now. This paper here from, you see the figures here in this paper. This was quite a seminal findings. What they did in this study, I'm probably going to go deeper into the science than anybody expected at this meeting. What they did was connect two mice together. This is a young mouse in yellow and an old mouse in gray here. So this is called heterochronic parabiosis. The mice then share each other's circulatory systems. And what happens is this sharing of the circulation between the animals changes the biology in the other animal. In particular, that younger animal, even in the brain, we're connecting circulatory systems, but we see effects in the brain. We see a more of an aging phenotype in that younger animal. We see, change the figures here, a neurogenesis, an aging of the stem cells and the ability to make new neurons. You see that reduced in the young animal. So the circulatory system is affecting the sort of aging biology in the brain. And what you see on the right here is this has now been shown across multiple organs through heterochronic parabiosis, through plasma exchange experiments. You can change the aging phenotype of a multitude of organs, brain, muscle, liver, the heart, bone. We see across multiple organs that the plasma proteome is not just a marker of effects, it actually has functional consequences on aging biology. So now we have we have this way to actually change a multitude of biologies across a multitude of organs, and really this potential therapeutic opportunity. And I want to show you one experiment, how this really is happening. So these are experiments that we've performed in our labs to look at can you take plasma into an old animal. We're not doing this in humans at this stage. I'm going to give you some human data in a little bit that's more disease-related. But can we take an old animal? So these are about 2-year-old mice. If we compare this to sort of a human age, it's 80s to 90s, very roughly here. And what we do with these mice is train them to test their memory. So this is called a Barnes maze. There are multiple holes in this maze, and the holes circled in red are ones where the mouse can escape. We put the mouse in, we train it to where this escape hole is. And what we've done here, the mouse on the left, we've infused with saline for 2 weeks. So this is an old animal, just infused with saline for 2 weeks, and we've trained it where this escape hole is. I'm going to start the video. And this mice, even though we've trained it, we've shown it where its escape hole is, it's wandering around. It wanders around. It's really not remembered very well where it's going, its location on this maze. On the right, this is a litter-mate animal. It's a brother of the one on the left. The only difference here is we've infused this mouse, it's a 2-year-old mouse, with 18-year-old human plasma. And already this mouse went off to the right sort of zone. It was recognizing the cues around this maze and actually it got to the right part of the maze and has very quickly gone to its escape hole. So you can just putting 18-year-old plasma into a 2-year-old mouse actually improve the cognitive function, improve this systemic function of the animal, so you can improve overall biology. The video is not going to quite get there, but I promise you the mouse on the left does actually get there. It's going to get there right now. So people can learn, but we can actually change the composition of the plasma proteome and speed up cognitive processes, shown here. So this introduction here is getting us to the point that we can understand from the plasma proteome aspects which can influence aging processes. And what we want to do is now really put science to this. What is it in the plasma proteome that influences aging? Can we take advantage of plasma as this master communicator between organs and have this broad impact? We signal between different systems and organs. We factor in onto what changes as we age, and really then build these therapeutics that can increase health span and get to these goals of having much healthier life now through an intervention. So as we're trying to decode this, the plasma proteome, we're looking for what are the proteins that have biological impact and that change with aging. We've called these chronokines, chrono for age and kines with the functional component. And in a very simplistic terms, there are many proteins that go up in levels. These can be perceived as ones which are negative and ones which we may want to reduce to actually get back to a younger overall sort of aging concept. And those that go down with age that we may want to increase, so more normalized to a younger level. And we have various ways that we can go about this. We can reduce or antagonize these detrimental proteins using standard small molecules, antibodies, or we can increase with plasma fractions that I'm going to show you, or supplementing proteins, and try and get this balance of biology back again. So I'm going to show you now that this really is doable, and we've made some good impacts in these approaches. What we need to do is understand this well. This is not a small question here. The plasma proteome is complex, it is big. We're now able to look with very deep profiling at about 8,000 proteins on a routine level. What you see is that there are proteins that change in waves over aging, that, as you see in the figure here, blue is lower levels, yellow higher levels. Different proteins are changing at different rates over aging. So we start understanding these profiles and see, yes, you actually have an aging proteome change that you can tap into. One of the big challenges we have is you don't right now develop a therapeutic for aging. We have to work through indication areas. And so we can start now matching, these are the proteins which change with aging. They're also connected to diseases. We don't have to dissociate completely. These are proteins that are connected to aging and specific diseases. And we've got a number of data sets that we built in subjective cognitive decline with samples from Dr. Boada's group, in Alzheimer's disease, Parkinson's disease, a range of areas. But there are also now data sets that help you understand across a whole range of different disorders. Here are proteins that change, those that are connected with aging, and give us tracks to be able to follow the aging disease connection. So what we do, what I've shown you so far, is we look with proteomics. Here are the proteins that change. We use, it's complex. We have to go into the world of AI to actually prioritize these proteins, understand what could be the drivers, and then do in vivo and in vitro validation that then deliver candidates so that we can actually modulate this biology of aging. I'm going to show you a couple of examples here. So going back to the biology of aging, we can break down the biology into these different mechanistic areas. We can look at what changes with aging. You see here in red are proteins that increase with age, blue those that decrease, and how they scatter across these different mechanisms. So I'm going to show you two stories, one around plasma fractions. These are the GRF6019 and GRF6021 stories, where we're able to modulate multiple of these beneficial chronokines and have effects. And I'm going to take one example, AKST4290, where we can go into a single chronokine, inhibit that signaling, and affect in this immune aging area. So first of all, on the plasma fractions. The world of plasma fractions takes plasma from donors, fractionates through a manufacturing process using temperature, pH, etc., to develop products. And these products are used, factor VIII for hemophiliacs, IVIG, albumin. You're going to hear a little bit more on that later. This is an establishing. So we hypothesize that you could go from these concepts. There's something in plasma. Can we find the more anti-aging, pro-cognitive in particular, fraction of plasma which could have benefit? So we've tested a large number of plasma fractions. I'm going to show you data here with one of these called GRF6021. And what you can see when we infuse this plasma fraction, it's still a mixture of hundreds of proteins. There are about 500 proteins in this plasma fraction. We can improve the biology of aging even in the brain. I'm just focusing here on the brain because this is the sort of the most differentiated that we think of from the plasma proteome now to the brain. We see increases in neural activity when we look at immediate early genes here. We see reduction in inflammation. This is reduction in microglial markers in the brain. Increases in synaptic connectivity. And changes in neurogenesis, formation of new neurons in the brain. And this happens with different timescales. So from hours to even months after a plasma fraction treatment, we can see these benefits. So you're able to trigger with a plasma fraction changes in biology that we connect with aging, even in the brain. We can also connect that this biology is actually having a mechanistic impact. We use single-cell transcriptomics. So the brain is complex. It's composed of a large number of different cell types. We can actually separate out these different cell types. And you see here a figure where we've separated out astrocytes, neurons, endothelial cells, and we can now look at the transcriptome, what has happened in each of those individual cells. And what you really see remarkably in the middle here is when we look at the capillary cells, so this is the brain microvasculature, with aging, we see a very specific profile of changes. There are some downstream signals that are decreased, some that are increased. When we treat with GRF6021, we can actually reverse that biology of aging in the brain microvasculature. So we are really having this impact with a plasma fraction treatment on aging biology. It is feasible. And so we can start connecting. Here are the proteins that are in the plasma fraction, here are the transcriptome changes, and start rationalizing what is it about plasma that is having these effects. And it really works. So we've got a plasma fraction here which improves cognition. So this is graphing those videos that you saw, the time of escape on a maze here. An old mouse takes a long time to escape. It learns, but it learns slowly. Treat with a plasma fraction, you can get back to the function of a young animal. And when we look at neurogenesis, this process of developing new neurons in the brain, we can actually, with a plasma fraction here, have an even enhanced activity compared to whole plasma. So we're able to enrich the factors which are important in affecting the biology of aging. And we have taken this now into the clinic. We've done a number of clinical trials I'll show on the next slide, but this is very quickly in the area of Parkinson's disease dementia. We've taken this plasma fraction, GRF6021. We have dosed patients. We had, appears that we have 5 days of dosing, and then 3 months later, we do another 5 days of dosing of this plasma fraction, a randomized placebo-controlled study. And what we saw in this now in patients with Parkinson's disease dementia, we see improvements in cognitive function, meaningful differences between the treatment and the placebo group, safety and tolerability of this, and we have superiority compared to parallel studies that have been done. So this is looking at cognitive function, a verbal fluency test here. We have an increase in verbal fluency, and this compared to drugs which have been tested in similar paradigms, rivastigmine approved and donepezil, the Alzheimer's medication. We have improved function. So we're on this path to taking these plasma fractions forward. And what we think of is very broadly here. We think of this towards Parkinson's disease dementia in that case, but we've done trials with related plasma fractions in Alzheimer's disease. These are published studies. I'm happy to share these publications to anyone interested in mild to moderate Alzheimer's disease, severe Alzheimer's disease. And we are just finishing up a study now in recovery from surgery after hip and knee replacement surgeries in elderly people. Thinking about this concept it's more broad than just Parkinson's disease, Alzheimer's disease. It's a systemic biology that we're changing. I'm going to just show you now 2 or 3 slides on going from a different angle. Can we move away from a plasma fraction into now taking some of the individual components and targeting those? So we have a lead candidate which is targeting this inflammaging area. So what we found in plasma was an increase in a protein called eotaxin, also known as CCL11, increases with age in quite a linear fashion. When we think about CCL11, we think about what does it act on? Its primary receptor is called CCR3, and so we can now block the interaction between eotaxin and its receptor and see if modulating this biology has effects. We always think back to the connections with diseases of aging as to where to target our clinical development. And there are genetic linkages to age-related macular degeneration, this eye disorder. And in Alzheimer's disease, there have been a couple of studies now that have shown that there are mutations in the genes for eotaxin and CCR3. So we now have some connections which indicate, yes, you have a biology here that's relevant to individual diseases as well as the aging process. So we've looked in the eye as a first part. We see when we treat with this compound, we do have effects on inflammation and infiltration in the eye. So in the human condition of age-related macular degeneration, you see here an infiltration of immune cells into the retinal pigmented epithelium, the choroid layer, associated with AMD. So this is in the human case. This immune cell recruitment, infiltration is important. We can develop a model in the mouse where we can treat with sodium iodate, and we can induce inflammation, we can induce retinal degeneration. And then when we treat with this compound that's interfering in this process, we can reduce inflammatory cytokines, you see on the left, and then infiltration of cells into the eye of a mouse. So we're building up this story in a preclinical model. We don't think single indications. We think that this is relevant to multiple indications. We've gone into studying Parkinson's disease, which has big neuroinflammation components. We've worked with a model called the alpha-synuclein transgenic mouse. In this mouse, again, we can treat with this compound that blocks this interaction, and we can see improvements in function. So in a variety of behavioral tests, we see that the transgenic mouse slips more on a beam. It can't hang on to wires. It has less motor coordination. But when we treat with the compound, we're reversing some of those effects. So we don't just think the eye, we don't just think the brain, but we see this broader effect. We see effects on neuroinflammation, as you see on the right. So again, we go from the mouse into human studies. We have now run a number of clinical studies with this compound in the eye. Treat with the compound. This is daily for 6 weeks total here. We see an improvement in vision in subjects. So this is letters read on an eye chart. So you're seeing here 7+ letters improvement. This is more than a line when you read an eye chart. Improvement in visual function. So what we see in the mouse, we're actually now seeing in the clinic as well. You can take on this biology and really make progress. So again, we've thought here quite broadly. Here's a mechanism of aging. We have trials completed in age-related macular degeneration at the top here in 2 groups where we saw these benefits, much you've seen in that slide. And now we have studies in Parkinson's disease, which we'll be reporting at the ADPD meeting in March here in Barcelona, and in bullous pemphigoid, a dermatological disorder, which will be announced soon as well. We can think broadly. The science is there and coming along. So hopefully today I've shown you what we're doing here is reality. We can approach the science of aging through decoding the plasma proteome. We can influence the aging process and have potential therapeutic approaches. It is scientifically rational, this approach. We have quite profound systemic effects, and we can do this through plasma fractions. We can do it through individual targets, and we can really develop transformative therapeutic opportunities. So there is a reality to this science of modulating aging that we're tracking through in a rational way. I'd just like to acknowledge, this has been a team of multiple people at Alcahest and colleagues at Grifols. We always have to thank the patients and the families, the caregivers, and the investigators in the trials that really enable us to translate this science forward. I'd be very happy to be in touch more on all of this science in the future. So thank you very much.
Thank you also, Stephen, for that presentation. I think we started the presentation with the philosophy and the socioeconomics of aging, but now we've heard a lot more about the biology of aging and also some really interesting approaches of how we might combat almost aging through plasma exchanges. So I also have a very quick question here because somebody actually used the question, and it's a question for clarification. And the question is it just the transcriptome which is important in aging, or also post-transcription changes, e.g. glycation and oxidation?
There's a huge biology which is impacted here, and we certainly think that the glycation, post-translation modifications of proteins are important. I think that Dr. Barda will have some data and some story there. This is a complex biology. It's not just absolute levels of proteins, it's the quality of those proteins as well that matters.
Okay, great, thanks. And that's also a reminder to anybody that has any kind of questions, please use the app, and we'll get back to your questions at the end of the session. But after this, it's over to Merci Boada, and she will be updating us. So we've heard about the philosophy, economics of aging, we've heard about the biology of aging, and the plasma proteome. And now we are hearing from Merci Boada, and she's going to give us an update on a really innovative Alzheimer's treatment, which is plasma exchange with albumin replacement, or AMBAR in short. Merci, the floor is all yours.
Okay, good morning. Thank you for offering me the opportunity to talk about this beautiful project that I have been involved for more than 15 years with the team of Grifols and the team company, and my first gratitude for the Grifols trust in our institution and also trust in me. Well, maybe this last lecture, it's moved from the big picture, like Professor Scott is talking about aging. No? I'm sorry. Gracias, it's not back, eh? Thank you. He's talking about the big picture, is how it's important to predict a wealthy life, wealthy to reach to the at the end of our life in the good shape in aging. Secondly, it's the how it's the plasma, how it's the blood, and how it's the proteins increase or decrease and change our metabolism. And finally, how all this knowledge focus in one treatment. Because one of the things that it is important for us is to treat patients that his brain is not working well. I want to talk today about how we move from the hypothesis and the knowledge of pheresis to Alzheimer's disease as a new treatment. Of course, I want to talk about AMBAR because it's the core of my lecture, to talk about the AMBAR, the reasons to do, and the results. Also, beyond AMBAR, non-clinical research. At the end, the Therapeutics Pheresis Unit on ACE is the AMBAR Center that we are working in the real world with the AMBAR study, with applying the AMBAR study. Okay, as Dr. Ascott said and show you, we have a wonderful brain full of connections, but so pity, this wonderful brain lose connections. And the question is what it means, and the question is how we treat it. The first one, it's very easy to answer. Dementia, a disease. The other is a big, big challenge in order to treat this really catastrophic disease and reach healthy brain at the end of our years. Well, what is the link about pheresis and the Alzheimer's disease? And that is the topic, the main topic from this panel. The AMBAR study is a new paradigm for use the pheresis in a complex disease because as Stephen has mentioned and repeat in the different slides, I summarize this one. Alzheimer's disease is a biological complex disease because have a multifactorial etiology, but it's not only biological complex disease, it's a social, social complex disease, of course, no? And the other is a genetically complex disease because it's not depend for only one gene, it's depend for one polygenic risk score, and that is very difficult in order to find a very precise treatments or personalized treatment that is our future. But one of the main important causality of Alzheimer's disease is aging. Aging with this complexity. They have one hallmark, it's the burden of the same proteins, misfolded proteins, like beta-amyloid, that is the main protein that we consider the most prominent cause of Alzheimer's disease. This other is neuroinflammation, oxidation, the pathology of the microvasculature, genetics, of course, the linking from neurotransmission in order that one neuron talk on the other, and that is focused in the synaptic space, energy from the mitochondria have a dysfunction, will lose energy, and to produce and to maintain and keep the homeostasis and this brain, and all this one we're going to treat. If you focus in this slide, do you know that the neural death, it's not only for inflammation, for beta or for some defects in the vascular, it's a multifactors, and that it's the huge causes, the huge elements that seems to be impossible to treat Alzheimer's disease with only one drug and only one approach. AMBAR is another approach that is focused in order that offer me the possibility to talk about AMBAR is one octopus treatment. I like this one. How we understand one of the proteins, the most abundant proteins in the plasma, play a role in the Alzheimer's disease, that it's important. Do you know, Steve knows very well that albumin is the most abundant protein in our plasma, even in the plasma, and also in the CSF, in the central zinc. The other thing is really albumin maintains the microvasculature or the intravascular volume. It's the main transporter from the fatty acids. It's important for drugs, like the drugs for Parkinson's disease, is from another's metabolism, and also for A-beta. The other is it's the main extracellular antioxidant and have immunomodulatory effects. But sometimes albumin becomes worsening because it's under oxidation and glycation and translational modifications. Okay. And how albumin, these characteristics, takes relevance in Alzheimer's disease? Because when the actions and the functionality of the albumin keeps the A-beta affinity, 90% it's bound. A-beta is bound in albumin because inhibits the A-beta aggregation and prevents the A-beta internationalization in the neurons. But when albumin is altered, this albumin altered is increased in AD for the process of glycation or for nitrotyrosinate. This albumin altered shows a lower affinity to inhibit A-beta aggregation, and in an animal model, when we infuse albumin in the brain of this animal model, they reduce the amyloid burden and the brain inflammation. And all this one is our background to plan and to start the hypothesis of the plasma change with albumin replacement. And as you can see here in this diagram, that it's trying to mobilize from the central zinc to the peripheral zinc, and that it's one important question that it's very difficult to resolve, that what is the play, the role play of the brain-brain barrier. Peripheral sequestration of amyloid species through the plasma change may achieve the equilibrium from the peripheral brain A-beta and to promote one benefit of flows from the central zinc, brain on the CSF, if you consider this one, to the peripheral zinc. That is the hypothesis that we are managing to plan all the development of the plasma change with albumin, and the final step was AMBAR, will be AMBAR. What about the AMBAR study? What about the AMBAR study? What the AMBAR study is Alzheimer management by albumin replacement and combine two strategies, one mechanical strategy, another to replace for the most abundant protein in our body, the albumin. Talking about the mechanical remove, in the mechanical we remove plasma with the toxic amyloid protein, but another things, cytokines, antioxidants elements, and neurofilaments, of course. And we replace with young plasma. As Steve was talking about this, was young plasma. And young plasma keeps the old functionality of the albumin, and this new albumin provide the antioxidant properties and binding, and to keep the binding capacity. That is the first strategy, as you can see in this slide, to understand the AMBAR project and the AMBAR treatment. How about the program overview? How long we are treating and studying and test and promote clinical trials in order to reach the final phase 2b/3 AMBAR trial. First one, it's a pilot in order to understand this kind of the treatment, this approach could be feasible in Alzheimer's patients, because Alzheimer's patients, it's not easy to manage. Maybe it's more difficult to manage the mice and to manage the animal model. It's quite difficult because there are a lot of conditions that interfere in this treatment. Second one, we design a pilot extension in order to it's possible to reproduce the results of the previous study. The phase 2 is a confirmatory study in order to collect all the results from the previous pilot and to understand the safety and efficacy in this phase 2. And finally, we designed the AMBAR study is accepted like a phase 2 and phase 3 in Europe clinical trial. If you are interested to know in more detail and get more information about all overview program, it's more than 15 years, nearly 17 years, we have the opportunity to read this paper that I was considered the more relevant in order to understand, first one, the efficacy and safety, second one, to in detail the study program and the progress. In the 3 paper, we can see all results. Today, I don't show all the results, but the most significant results. And finally, the recent paper is about the neuropsychological, neuropsychiatric, and quality of life assessment that it provides the results of AMBAR study. Go ahead. What about the AMBAR design? AMBAR design is designed in 2 different periods, in 2 stages. The first stage, named the intensive period, is when we use the total plasma change therapy, and the patients we treated with 1 treatment, total plasma change, every week during 6 weeks, for 6 weeks. And we replace albumin 5% from 125, 150 grams with 1 sham. And in the second period, maintenance period, it's during 12 months, the patients are treated with a low volume plasma change 1 monthly. Another picture, and this is schematic AMBAR flow chart, you can see here the total plasma change, 1, 2, 3, 4, 5, 6 treatments with albumin 5%, with a sham. And during the low volume plasma change, we compare and contrast 3 arms that we introduce the immunoglobulins, high level, high dose with albumin and immunoglobulin, 40 grams, 20 grams. The low volume with 20 grams of the albumin and 10 grams of immunoglobulins. And one of the active arms, it's only use the low volume of albumin without immunoglobulins. And you can see in purple the sham. In the symbols, you can understand how many complementary tests and tasks it's involved in the flow chart of the AMBAR. It's biomarkers, neuropsychology tests, lumbar puncture, 1 here in the baseline, when one finished the intensive period at the end of the treatment, FDG and MRI. It was not so much complex clinical study, even though this schematic seems to be complex. It's easy, 1 period and the second period, and we compare 3 arms versus the placebo arm. How it's important to talk about the inclusion criteria, the patient's eligibility, because patient's eligibility it's included from 55 to 85 years. And we include patients without biomarkers, not using the ATN criteria. we include patients by phenotype. And phenotype because our deal was mild to moderate patients, one MMSE from 12 to 26. Of course, must be stable in the current treatment and have the MRI, current MRI lasted 12 months, and as all clinical trials in Alzheimer's disease, we need one partner and we need one caregiver in order to take opinion about the process and improvement on the worsenings. What is the patient and the size disposition? Well, the size was 41, divided in Spain, 19, and the US, 22. We reached 500 patients, but the most important thing here is an underline in this box, is only 30% it's screening failures. 222, it's patients that we can evaluate. 7 to complete the treatment, but the most important thing here, and is you think about what is happening with the placebo group, the placebo group reached the 80% to complete this treatment. Run through very fast from the baseline characteristic. I only use the MMSE compared to placebo group to the total sample, and the other score to the total sampling, and you can see in the different colors the high dose, low dose, and low dose without immunoglobulins. But the most important in terms that we make one approach from the novel clinical trial that they use the ATN like a principal criteria to involve patients in these clinical trials. That is one first. How is the proportion in one genetic risk, very well known risk factor in Alzheimer's disease, ApoE carriers or non-carriers, ApoE4 and no. And if you go on here, as you can see here, it's very well balanced from the placebo and the total sample. And in terms of the signature of Alzheimer's disease, A-beta 42, we have 3 portions versus 1 in order to divide this sample. 75, that it was a normal amyloid, versus 25, it's normal amyloid. And remember that Alzheimer's disease is a continuum process, and even though that we detect in the phenotype that this patient exhibits all the symptomatology from to be included as probably Alzheimer's disease or possible Alzheimer's disease, part of them, they don't reach a significant ratio in the biomarkers like A-beta 42. Follow on to the results. I'm sorry for my team in Grifols because, as you know, we can manage more of this data. But Tony, I only use the most relevant in order to reduce 15 years in 20 minutes. The most important results for us that it's mandatory from FDA is the co-primary results. As you can see here, the function in these patients, that it is important, cognition and function, and that it's important for Alzheimer's Europe and for Alzheimer's disease, because I want that the patients have capacity to live alone, that it's a real endpoint in terms on social cause or indirect cause. The ADL, as you can see, we reach a p-value, significant p-value, and effect size, the more than 50%, or the less decline more than 50%, and the difference from the treated patients and the placebo reach 3.5. But in terms of cognition, it's not so successful. But our trends shows a very good trends, because even though we don't reach here, we don't reach here the p-significant value, the effect size is we compare cognition, global cognition in this global battery from the purple treated patients and from blue is the placebo group, we reach 60%, 66% in the less decline on the effect side. But move on to the key secondary results. That it's important. I want to emphasize that in the new clinical trials, on the more recent clinical trials, CDR sandboxes was accept as a force or the main objective in the clinical trials. And I want to emphasize that in our AMBAR study, the CDR sandboxes is a key secondary results. Change the idea if at the first when design AMBAR, we change the principal endpoint for this secondary endpoint. What has happened in the CDR? In the CDR, we reach a p-value significance, robust, no doubt, and less decline, more than 70%. That is incredible if we compare the other clinical trials focusing disease-modifying treatment, because don't reach these spectacular results in the CDR sandboxes. But if we compare this one in another measure to show the functionality, the activities lately given, instrumental, the executive daily activities, daily living, you can see here that the appreciation, the information from the partners, from the caregivers, it's that this group, the purple group, the treated group, is keeping in one effect size of 100. That is astonishing. And that is my opinion. I'm sorry, but under my opinion, that is astonishing. How about the AMBAR safety? Because we use one device, seems to be very difficult to manage. It's very difficult to manage for the plasmapheresis. Maybe it's very difficult to manage because we are treated Alzheimer's disease patients, and not patients we have their capacity to take decisions. I want only this one, underline this one. Less than 2.5 in terms of the catheter reactions. I talk about, if you have any questions about the catheter, hypotension, muscle spasms, but here, syncope, anxiety, nausea, paresthesias, presyncope, it's non-reach 1% of the adverse effects during the process. Go ahead. That my conclusion here, the conclusion of this and this slide is, in terms of safety, plasma change is safety. It's very well expressed a very well tolerance, and is feasible in patients with mild to moderate Alzheimer's disease. Beyond AMBAR, my third key points, I want to talk about the non-clinical research, in order how it's our background and how it's that we are producing and create to confirm our previous hypothesis and follow all the exposition that Steve was talking about, how appears in the aging people, aging animal models to using different proteins, and how it's these benefits. The first one, we are confirmed that patients under treatment modify the levels of glycate albumin and preserve a good albumin to keep their functional actions. In terms of lipidic, the lipidic profile, as you know, is linked to the lesions in the vasculature, and it's promote the microvascular disturbances, you know here that they modify the levels of the cholesterol and the triglycerides during the more than in the intensive period than in the maintenance period, but both decrease these levels, also in apolipoproteins, and we believe that this action is to reduce the vascular risk factor in the patients treated. But how is the important here, talking about the biomarkers, talking about the E-beta42, and talking about the total tau on the p-tau, when we consider that both proteins, that's both milfolded proteins, it's the core. It's one of the most important core in Alzheimer's disease. They keep maintenance in beta. It keep maintenance in beta. Also, in this group seems that beta can to be modified, and of course we have a good results with the p-tau and the phospho-tau. And at the end of this non-clinical study, you can to say that plasmapheresis interferes in the positive way in the Alzheimer signature using only these two markers, beta and tau. And the most recently, that is the poster that is exhibited during this week in the CITAT in Boston. In this poster, it's talking about the neuroinflammatory effects of the plasmapheresis. And if you can see here a little more, because I copy the poster, and I'm sorry and I apologize, because could be you can to see very well this slide. But if you compare here the amount of the proteins, the seroinflammatory biomarkers in the mild and the moderate Alzheimer's disease, that is the most common and the most important in terms that how influence the inflammatory biomarkers in the sero. You can see here that they are a big difference or the different behavior when we talk about the patients treated and with the placebo group. All these non-clinical research confirm, and it's a good background in order that our previous hypothesis to mobilize beta from one and the other, from the one thing to the other, could be effective in base of this all non-clinical research based on AMBAR study. In summary, but not the end, only in summary, I'm sorry. In summary, we have seen the AMBAR results offer a clear and evident clinical benefits in mild to moderate Alzheimer's disease. Mild to moderate Alzheimer's disease is at the end of the disease, mild no, but moderate, yes. And if you compare this one with the others components that is included in modifying disease treatment, we have unclear and evident clinical benefits using AMBAR strategy. AMBAR results are backed up by a solid scientific basis, as you can see in my previous slide, and in terms of the tolerability, because that is important in Alzheimer's population, in terms of tolerability and safety, AMBAR has proven to be safe and feasible treatment to apply. To apply today? To apply tomorrow? That is your question. To apply today? To apply tomorrow? I talk with you about how to do in the AMBAR Center today. Oh, I'm sorry, back. That is 2 slides, one and video, in order that to understand and to share with you our first patients treated in Alzheimer's Center as a real-world AMBAR therapy. Thank you. And that is a video, it's not so long video, that shows the patient journey, and at the end, I want to share with you some data about the current data in the AMBAR Center. Finally, I thank your time and your attention, And thanks my partners and the chairman and the Grifols company and the beautiful team led by Tony Paez that I was involved when I was a young neurologist. Thank you very much. And that is the video. I'm sorry, that is the video. And later the results. Maybe I talk the results in my seat. Thank you. Go to. And the patients treated. My last, no. I have one other slide. You come back, please? Thank you. Because in this slide, I try to summarize the real world evidence in AMBAR Center. And the first patient was treated in the 26th of May this year. And we have treated 65 patients, 32 females and 24 males, and with mild to moderate Alzheimer's disease. And 2 patients drop out, one for the cancer prostate, the prostate cancer, and another because the family discontinued the treatment, the family. And now we have 12 patients that it's nearly reaching the 5 and 6 total plasma change. And 7 of these, it's entering in the maintenance period, low volume period. 5 is pending to be scheduled during this year, this month, and the next December. And we plan to have the first analysis when we collect the data for the 50 patients that we had finished the total procedures. Something more about demographics is the middle age is 21 years, and the MMS-E is around 19, between 9, because we have patients with disturbance in language, to 28, and well, I have not more data at that moment to share with you, but I am open to and free to answer your questions. And if you are interested to visit our center, the AMBAR Center, please contact with us, and it will be a pleasure. Thank you very much again. Thank you.
Thank you very much. Merci also for this presentation. I think this gave you a really fantastic overview of a very innovative treatment approach. You got some very good clinical trial results, but also you saw how this could fit in a kind of real-world setting. There's also a number of questions. I'm just going to see whether Andrew is also able to join us, because we have a few questions that we will be able to touch upon. And maybe I'll start with Andrew, because one of the questions I had for you is research funding is very much focused on individual diseases, and kind of your view of attacking aging rather than individual diseases. Would you have any kind of recommendations or advice to research funders, but maybe also for pharmaceutical companies? What would be the targets and how should that be funded?
Yeah, and obviously I can only talk about the economics. There's obviously the biological issues, but what my results show is, first of all, given the probability of the young becoming old now in high-income countries, the biggest health challenge is to age well. There is this cluster of age-related diseases, and of course, if we can find ways of changing the pathways of aging, the effect gets magnified because you're summing across several diseases, and that leads to those synergies that I was talking about. So the obvious implication is to increase the amount of funding that goes into trying to understand the biological pathways of aging, for sure. What the biology says we don't yet know. There's a lot of progress being made, but yeah, absolutely to reallocate it. Let me also just add one other thing from a sort of non-pharma point of view, which is that anything that improves how you age has a really big payoff. And that's exercise, that's education, it's quality of housing. So I don't think it's just the pharma and science funding organizations. I think it's government as well who need to sort of think less about intervening for individual diseases and more about things like public health, which would also have a huge impact.
Thank you very much. And then the next question that I have is one for Dr. Braithwaite. Braithwaite. And this is a question about shortages of immunoglobulin. Is something that is currently a real problem, and how do you envisage solving that, or how will that impact the kind of research and therapeutic approaches that you are investigating?
I think we're understanding a wider biology beyond just immunoglobulins. It's not all about immune response, although that is important. I think longer term, we have to think about how do we scale all of this that we do. The population getting older is huge, and as the more we can learn from recombinant approaches, which can take this on in the long term, we're going to need to go there. So as we've got to understand the molecular basis, we've got to develop things which are truly scalable, as well as exercising more and eating better as well. I certainly agree, but we've got to think on that larger scale.
Then another question that I have here, and I think it's also for you, and this is that according to Susana Casas, is that the plasma of centenarians seem to have different characteristics to those of elderly with chronic diseases. And then the question is, do they just have younger plasma than those adults with chronic disease?
Again, it's all about molecular composition, and we shouldn't just think of everybody at one age is not the same. It's biological age of different people is very different. So chronology and biology of age do dissociate. We have an awful lot to learn from centenarians, people who exercise strongly later in life. We can understand that molecular basis and then sort of build that into our thinking. So I think it's a much bigger thought.
I have a question for
you. Absolutely, Natalie, go ahead and ask me a question.
Hello, we are talking about for a long time that Alzheimer's disease is a good model. Understand as a model, as a biological model, in order to understand aging and understand how is transforming the proteins during this continuum. Because your job, your work and your investigation with us is modulating healthy people with the promorbid, his mild cognitive impairment, because in mild cognitive impairment, I don't know if you are agree with me, that is the big topic in order to treat, because it's difficult to treat non-clinical patients, not asymptomatic, but mild cognitive impairment offers the possibility to detect some defects. And they are moving through all the continuum Alzheimer's disease and reaching the moderate and very severe. And you understand that all this fun And all this exhibition of the proteins can offer the possibility to modify the process, not only aging, because it's include aging, it's the process of Alzheimer's disease.
Yeah, I think looking in much earlier stages of every disorder is important. We've got to look for earlier points to go in, which means our therapeutic approaches need to be as safe as possible as well. I think this is a really, really big hurdle, which is why data from AMBAR is exciting as well. Safety and tolerability is strong, which is why a lot about what we've learned from aging are things like metformin, things that have been on the market, studied through other indications first, and then we learn the broader aging components later. So yes, we need to intervene earlier. And exercise, yes, we should be doing that all the time. But as we think about more therapeutic approaches, safety is going to be very key.
To move to the prediction and prevention.
Yes, yeah.
Okay.
And then I have two more questions, and then I think we need to wrap up. And one is a very specialized question. So this one is from Stephen Fava. And the question is, if albumin treatment can mobilize amyloid proteins from brain to the periphery, is there a risk of increasing aging of peripheral tissues, for example, increasing pancreatic amyloid?
Let's see. Yeah.
Why you question?
I can promise you this is not a question that I have come up with myself. So we have people in the audience or connecting online that are highly specialized.
Yeah, you are talking about if to mobilize albumin and proteins from one peripheral cell to the central cell, it's improving and could be worsening.
So I think if I understand this question correctly, or maybe correct me if I'm wrong, but it's really saying that...
you compare parabiosis with our treatment, no? That it's the question. I believe that really, when we increase the capacity, first one, in the mechanical performance, because we clean one plasma full of toxins, and we replace with the same volume with one protein, is a young protein, albumin, have the capacity to constantly, like a vicious circle, constantly to clean the peripheral thing. And I believe that it's impossible to worse the brain conditions, because that it's really a benefit of the homeostasis, the homeostasis on this brain. That is the finality of the AMBAR study, but I believe that we are confirmed that this hypothesis is make us in a good position to get the benefits in the brain.
So, and I think maybe there was a question around potential side effects that by mobilizing amyloid proteins from the brain, that might have an impact on the pancreas. You wouldn't think that would be a danger?
That until now it's not.
And I think with that, I'm also aware that we are 6 minutes over time. So for me, I think at the beginning of the session, I promised you a really interesting agenda. I promised you 3 fantastic speakers, and I think I haven't over-promised that, because I think we've learned an awful lot around the economics, the philosophy of aging from Andrew. We heard about the biology of aging and plasma exchange from Stephen, and then we heard a fantastic presentation from Mercé, really about an innovative treatment, giving hope to people living with Alzheimer's disease for a potential change and improvements in their lives. And really, my thanks go to all 3 of you. I've learned an awful lot in this session, and I really hope that you as an audience enjoyed the session as much as I did. But again, thank you very much for your presentations. And with that, I hand over to the lunch break that I hope that you're all going to enjoy. And before that, don't forget also to use the evaluation form and let the organizers know how much you enjoyed, hopefully, this presentation. Thank you very much.